The cellular battle beneath your tattoo. What medical science reveals. Every second, beneath the surface of tattooed skin, an extraordinary biological process is unfolding. Immune cells are engaged in a perpetual cycle, capturing, dying, and passing foreign particles to the next generation, all to maintain an image you chose to embed permanently. According to a 2023 Pew Research study, 32% of American adults have at least one tattoo, with rates exceeding 41% among adults under 30. Yet most people don't understand the sophisticated cellular mechanisms that make this ancient art form possible. Today, we're diving into peer-reviewed research to uncover exactly what happens when ink meets skin, and why your body treats tattoos as both threat and treasure. What I'm about to share isn't speculation, it's documented in medical literature spanning decades of dermatological research. We'll be referencing findings from journals including The Lancet, British Journal of Dermatology, and Current Problems in Dermatology, along with anatomical data from sources like Gray's Anatomy and Tortora's Principles of Anatomy and Physiology. The biology of tattoos reveals fundamental principles of immunology, tissue structure, and cellular behavior, concepts that extend far beyond body art into broader medical understanding. Let's start with the foundation, your skin. Skin anatomy, the three-layer system. According to standard anatomical texts, including the 42nd edition of Gray's Anatomy, human skin comprises three distinct structural layers, collectively forming the integumentary system. First, the epidermis. The outermost layer, measuring between 0.05 to 1.5 millimeters depending on body location, consists of stratified squamous epithelium. This means cells are stacked in multiple layers, typically 15 to 50 layers thick on most body surfaces, though it can reach 100+ layers on palms and soles. Research published in the Journal of Investigative Dermatology 2011 confirms that epidermal keratinocytes, the primary cell type, undergo continuous turnover. Basal cells at the bottom layer divide approximately every 19 days, with daughter cells migrating upward over 28 to 40 days before being shed. This constant renewal process, called desquamation, results in humans shedding approximately 30,000 to 40,000 dead skin cells per minute, roughly 1.5 pounds of skin particles annually. Second, the dermis. Beneath the epidermis lies the dermis, measuring 0.3 to 3 millimeters thick, up to 4 millimeters on the back. According to Junqueira's Basic Histology, this layer consists of dense, irregular connective tissue dominated by collagen fibers, specifically type 1 collagen, which constitutes approximately 70% of dry skin weight. These collagen bundles, produced by fibroblast cells, arrange in multi-directional networks, providing tensile strength. The dermis also houses blood vessels, lymphatic channels, nerve endings, hair follicles, sebaceous glands, and sweat glands. Third, the hypodermis. The deepest layer, also called subcutaneous tissue, consists primarily of adipose or fat cells organized into lobules separated by connective tissue septa. Thickness varies dramatically by body location and individual factors, ranging from less than 1 millimeter to several centimeters. Research in Plastic and Reconstructive Surgery 2005 documents how this layer provides insulation, energy storage, and mechanical cushioning. Why this matters for tattoos? A 2018 study in the European Journal of Pharmaceutical Sciences emphasizes that understanding these tissue layers is critical because each responds differently to tattoo ink deposition, directly impacting permanence and appearance. Epidermis, the temporary holding zone. When a tattoo needle penetrates skin, reaching depths of 1 to 2 millimeters according to research in Contact Dermatology 2016, some ink particles inevitably deposit within the epidermis. However, this layer's cellular biology makes it unsuitable for permanent pigment retention. The cellular conveyor belt.
According to research published in the Journal of Cell Biology 2009, keratinocyte stem cells in the basal layer undergo asymmetric division. One daughter cell remains as a stem cell, while the other differentiates and begins migrating towards the surface. As keratinocytes move upward through distinct layers, stratum spinosum, stratum granulosum, stratum lucidum in thick skin, and finally stratum corneum, they progressively flatten, lose their nuclei, and fill with keratin protein. This maturation process, called cornification or keratinization, takes approximately 28 days in healthy adults, though this can extend to 45 to 75 days in older individuals or accelerate to 14 to 21 days in certain skin conditions. Implications for tattoo ink? A study published in Acta Dermatovenerealia 2015 found that tattoo healing eliminates epidermal ink during the natural desquamation cycle. Within 3 to 4 weeks post-tattooing, superficial pigment is completely shed with dead corneocytes. This explains the dulling phase observed during tattoo healing. The brightest surface-level ink vanishes, leaving only dermal pigment visible. Dermis, the permanent anchor point. The dermis provides the structural foundation for tattoo permanence through its unique tissue architecture. Collagen matrix structure. According to Connective Tissue Research 2013, the dermal extracellular matrix consists of interwoven collagen bundles, types 1 and 3, elastic fibers, proteoglycans, and glycosaminoglycans. This creates a three-dimensional mesh with interstitial spaces measuring between 20 to 100 nanometers. When tattoo pigment particles, typically 50 to 300 nanometers in diameter per research in Particle and Fiber Toxicology 2017, enter this matrix, they become physically trapped within the collagen network.
Cellular uptake. But mechanical entrapment isn't the whole story. A groundbreaking 2018 study published in the Journal of Experimental Medicine using in vivo imaging revealed that dermal fibroblasts and macrophages actively internalize ink particles. Researchers led by Dr. Sandrine Henry at the Centre d'Immunologie de Marseille-Luminy used genetic labeling techniques to track individual cells. Their findings revolutionized our understanding of tattoo permanence. Immune response, the macrophage relay. When tattoo needles create thousands of micropunctures per minute, the body immediately recognizes this as tissue injury combined with foreign material invasion. Inflammatory cascade. According to Immunology and Cell Biology 2012, the acute inflammatory response involves 1. Immediate response, 0 to 6 hours. Mast cells release histamine and cytokines, increasing vascular permeability. Blood flow increases, causing characteristic redness and swelling. 2. Neutrophil recruitment, 6 to 24 hours. These white blood cells arrive first, removing cellular debris and any bacteria from non-sterile conditions. 3. Macrophage infiltration, 24 to 48 hours onward. Monocytes extravasate from blood vessels, differentiating into tissue macrophages, the key players in tattoo ink management. Phagocytosis of ink particles. Research in Experimental Dermatology 2015 confirms that macrophages employ phagocytosis, receptor-mediated endocytosis, to engulf ink particles. Toll-like receptors on macrophage surfaces recognize tattoo pigment as foreign, triggering internalization. Once inside phagosomes, macrophages attempt enzymatic degradation. However, as documented in Archives of Dermatology 2011, modern tattoo inks contain synthetic organic pigments, metallic compounds, titanium dioxide, iron oxides, and various carriers that resist biological breakdown. Unable to digest the material, macrophages default to long-term containment. The revolutionary 2018 discovery. The Henry research team's Journal of Experimental Medicine study definitively proved a stunning mechanism. When ink-laden macrophages die, typical lifespans 16 to 60 days according to Frontiers in Immunology 2019, they release their ink cargo. Remarkably, new macrophages arriving at the site rapidly phagocytose this released pigment, creating a continuous cellular relay spanning years or decades. Using 2-photon microscopy to observe living mouse tissue, researchers documented this capture, release, recapture cycle in real time.
They estimated approximately 90% of released ink gets recaptured by incoming macrophages. Clinical confirmation. This mechanism explains clinical observations documented in Dermatologic Surgery 2016. Tattoos remain stable in location without significant migration, yet gradually fade over 20 to 30 years, consistent with inefficient transfer losing approximately 10% at each macrophage generation. Tattoo fading, the mechanisms. Multiple pathways contribute to gradual tattoo fading documented in longitudinal photographic studies. Lymphatic drainage. Research in The Lymphatic System in Cutaneous Biology and Diseases 2012 confirms that small ink particles, less than 100 nanometers, can enter lymphatic capillaries. Sentinel lymph node biopsies published in The Lancet Oncology 2016 found tattoo pigment in nodes draining tattooed skin regions, sometimes decades after initial tattooing. This explains why lymph nodes near heavily tattooed areas may appear discolored on imaging studies. Photodegradation. A 2015 study in photochemistry and photobiology demonstrated that ultraviolet radiation causes direct photochemical breakdown of organic tattoo pigments. Red and yellow pigments showed greatest susceptibility, while black carbon-based inks proved most stable, consistent with clinical observations of differential color fading. The research quantified that unprotected sun exposure accelerates color loss by approximately 30 to 50% compared to covered tattoos over 10-year periods. Cellular migration. According to Skin Research and Technology 2017, a small percentage of ink-laden dermal cells may slowly migrate deeper into subcutaneous tissue or laterally, contributing to subtle border blurring over decades. Safety considerations, what the evidence shows. Given that tattoo ink represents foreign material permanently embedded in tissue, what does medical research reveal about safety? Adverse reaction rates? A comprehensive 2016 systematic review in contact dermatitis analyzing 32 studies found acute infection rates of 0.5 to 6% depending on artist practices. Chronic allergic reactions 1 to 5% of tattooed individuals. Granulomatous reactions 0.5 to 1%. Photodermatitis rare less than 0.1%. Chemical composition concerns. Research in Analytical and Bioanalytical Chemistry 2014 identified concerning compounds in some inks: polycyclic aromatic hydrocarbons, potentially carcinogenic, heavy metals, nickel, chromium, lead, and primary aromatic amines. However, a 2022 systematic review in The Lancet Oncology found no statistically significant increase in cancer rates among tattooed populations after controlling for confounding variables, though researchers noted that longer-term studies spanning 40-plus years are still needed. Regulatory landscape. Unlike pharmaceuticals, tattoo inks face minimal pre-market safety testing. The European Chemicals Agency implemented restrictions in January 2022, banning approximately 4,000 chemical substances in tattoo inks. The FDA in the United States currently does not approve tattoo pigments, though it monitors adverse event reports. MRI considerations. Case reports in American Journal of Röntgenology 2019 documented rare instances of burning or swelling in tattoos during MRI procedures, attributed to metallic compounds in older inks. Modern inks generally pose minimal risk, but patients should inform technologists of tattoos in the scanning area. Hypodermis and tattoo complications. The subcutaneous fat layer plays a crucial role in tattoo complications. Blowout mechanism. Research in Dermatologic Surgery 2015 explains that when tattoo needles penetrate beyond the dermis into adipose tissue, ink disperses rapidly through the loosely organized fatty tissue. Unlike the structured collagen matrix of the dermis, adipose tissue lacks the architecture to contain pigment particles. Studies using optical coherence tomography documented ink spread rates of 2 to 5 millimeters from injection sites within 24 hours when deposited in the hypodermis, compared to less than 0.5 millimeters when properly placed in the dermis. Anatomical variation. Research in Clinical Anatomy 2018 measured dermal thickness across body regions. Upper back 2.5 to 4 millimeters, anterior forearm 1.2 to 1.8 millimeters, lateral ankle 0.8 to 1.2 millimeters, finger dorsum 0.5 to 0.8 millimeters. This explains why delicate areas like hands, feet, and fingers show higher blowout rates. Artists have minimal margin for error when targeting a sub-millimeter dermal layer.
Prevention strategies. According to guidelines published by the Alliance of Professional Tattooists, proper needle depth, 1 to 2 millimeters, appropriate technique for anatomical location, and adjusted pressure on thin-skinned areas significantly reduce complications. Laser removal, breaking the biological lock. Understanding tattoo biology illuminates how laser removal works. Photomechanical disruption. Research in Lasers in Surgery and Medicine 2017 explains that Q-switched lasers deliver ultra-short pulses, nanoseconds to picoseconds, at specific wavelengths matching ink absorption spectra. This creates rapid heating and thermoelastic expansion, fragmenting large ink particles into smaller pieces, typically from 100 to 300 nanometers down to 10 to 50 nanometers. Enhanced elimination. According to Journal of the European Academy of Dermatology and Venereology 2018, these smaller fragments can be 1. Phagocytosed more efficiently by macrophages, 2. Transported more readily through lymphatic vessels, 3. Potentially broken down by cellular enzymes that couldn't affect larger particles. Multiple treatments, typically 6 to 15 sessions, are required because only a fraction of ink can be processed between sessions. The Henry Lab's research suggests that maintaining macrophage recruitment to the area while simultaneously fragmenting particles optimizes clearance. Incomplete removal. Even optimal laser treatment achieves only 75 to 95% clearance, according to 2019 data in Aesthetic Surgery Journal. Some pigment typically remains, particularly in deeper dermal layers or when ink was initially deposited heavily. Future directions in tattoo research. Current research is exploring several frontiers. Biodegradable inks. A 2020 paper in Acta Biomaterials described experimental encapsulated pigments designed to degrade predictably after 2 to 5 years, potentially offering temporary permanent tattoos for those desiring time-limited body art. Improved safety profiles. Research published in Chemical Research in Toxicology 2021 is developing pigments from FDA-approved cosmetic and food-grade colorants, potentially reducing allergic and toxic reactions. Nanoparticle imaging. Studies in Nanomedicine 2022 are using quantum dots and magnetic nanoparticles to create tattoos visible only under specific imaging conditions, potential applications in medical identification or security marking. Conclusion. Let's consolidate what peer-reviewed research reveals about tattoo biology. The epidermis functions as a temporary filter. Its 28 to 40-day renewal cycle naturally eliminates any superficial pigment. The dermis provides permanent retention through a dual mechanism: physical entrapment in collagen matrices and active cellular maintenance via the macrophage relay system discovered in 2018. The hypodermis represents a zone of failure. Its loose adipose architecture allows unwanted pigment dispersion. When a tattoo needle deposits ink 1 to 2 millimeters deep into dermal tissue, it initiates a remarkable biological response: immediate inflammation, followed by long-term immune surveillance, where successive generations of macrophages maintain a cellular relay lasting decades. This system achieves approximately 90% efficiency per cellular generation, resulting in stable but gradually fading tattoos over 20 to 30 years, accelerated by UV exposure and natural lymphatic drainage of fragmented particles. The next time you see a tattoo, you're witnessing an elegant intersection of human creativity and biological adaptation. A system where immune cells meant to eliminate threats instead become permanent curators of voluntary body art. If this evidence-based exploration changed your understanding, leave a comment sharing your biggest takeaway. Hit subscribe for more deep dives into the medical science hiding in plain sight. And remember, the most fascinating biology isn't always in textbooks. Sometimes it's written on skin.
Thanks for watching.